good afternoon, good evening, good morning again, and welcome everyone to what is a really exciting webinar in AVAC land here. I know it's only April, but we are very excited as we enter into a period leading up to HIV Vaccine Awareness Day, a day that is marked on May 18th each year, and it's been marked on May 18th for the last 20 years. And we at AVAC are excited and inspired and impatient that it is the 20th year in which this Vaccine Awareness Day is marked, and to recognize both the enormous progress and still the enormous urgency in the development of an HIV vaccine. We've decided to do more than just a traditional HIV Vaccine Awareness Day webinar to give an update on the latest in the science and advocacy related to HIV vaccines, because the field is at such an important point right now, sitting here in 2017. So over the next 4 weeks, in fact, there will be 5 different webinars, and you can see on the screen today is the very first, which I'll introduce in just a moment. But we're going to follow with 4 additional webinars, and the combination of the 5, we think, provide an incredible sense of the diversity, of the excitement, and of the still challenges in this incredibly important effort. In addition, there will be a range of other materials that people will be seeing through the Advocates Network and through other mechanisms to help people both understand where we are and what we still need to do in advancing the field. So we're delighted that people are on this line. In terms of today, I will say that the line is muted so that we keep the line clear as we're recording it, and we will post it and all the other webinars on the website as soon as each one is available, as well as resource materials that we have created for each of these calls. I would happily encourage people to be sending questions. If you are on the ReadyTalk interface, you can send questions through that. You can always email them at avac@avac.org. And once the presentation is done, I'll be asking questions and moderating that, but also happy to open the line, and I'll give instructions on how to do that so that all voices can be heard. As I said, over the next 5 or 4 weeks, we have these 5 webinars covering different aspects of research. And we're particularly excited to kick off this series with Maria Pau from Janssen Pharmaceuticals, which is part of Johnson & Johnson, the pharmaceutical company, to talk about one of the newer HIV vaccine programs, at least new to some conversations as that product advances through a number of trials, which Maria is going to describe in just a moment. It's an important and exciting program for a whole number of reasons, not least of which is that where Maria sits, not just in the Netherlands, but sits in a large pharmaceutical company. And when AVAC began 22 years ago as an organization, its very first report and effort was looking at the industry engagement, or lack thereof, in the search for an HIV vaccine back in 1995. And that was an advocacy issue in 1995, and it remains an advocacy issue in 2017 because, as I think everyone knows, industry's expertise and industry's resources can bring so much to bear in advancing an HIV vaccine program. And we actually saw that with things like Ebola vaccine development, which we'll hear about in the 5th of our webinars with Nelson Michael talking about vaccine development writ large. But industry's role is critical, and it always hasn't been there in the HIV vaccine field. So we are so excited that we can kick off this whole series with Maria representing the overall program that you'll hear about, as well as coming from a pharmaceutical company. The other important thing that makes this important, and as I said, each of the calls we have coming up over the next month will include resources to help advocates and others understand the issues at play. And as you'll see in a moment, Maria is going to be talking about the vaccine program that includes a Mosaic vaccine. And I won't get into those details because Maria can do it far better than I, but we have, and we will just be posting today, along with the recording of the webinar, this busy advocates guide to help understand this aspect of why mosaic vaccines are important in the field right now, and wanted just to flag that this will be up as a resource from the website, and others for each of the calls will follow as well. So that kind of sets the stage. Again, we'll go through the presentation, happily take questions via the ReadyTalk chat feature, as well as via email at avac@avac.org. And really excited for this conversation to kick off the first of this whole series. And again, I just want to introduce then Maria Pau from Janssen. And I know there are lots of names and pronunciations. Some people say Janssen, some say Janssen. It's all part of Johnson & Johnson, which I think people do know around the world. But previously, Maria had been with a smaller biotech that was called Crucell, that has really pioneered a range of vaccines, not just with HIV, that then became part of Johnson & Johnson. And so Maria has been part of this program and activity for many years, and we are so excited, Maria, to have you present today with an update on the Janssen Ad26 vaccine program. So with that, I will turn it over to you.
Thank you so much. Can you hear me?
Yes, I can very clearly.
Perfect. Thank you, Mitchell, for the great introduction and for the invitation to talk about our program today. And I will ask you to change the slide. Is that okay?
Yes, that's exactly right. We will do that as you go.
Great. So when I say our program, we can go to the second slide. And I immediately want to tell it's not Janssen only, right? Our program, I'm referring to all the collaborators we have currently. And I need to start, obviously, with our long-lasting collaborator, Dan Baruch at BIDMC. As Mitchell said, yes, I am working for many, many years, and I feel old but also proud at the same time with Dan Baruch and his team from the very, very beginning. So we worked on the discovery, on studying the prototype of what we now have in the clinic. And from start, pretty much from start, we also had great support from NIH with different grants, in particular substantial grants from the IPCAVD program. And soon after, a few years, we were joined by the military HIV research program with what I saw now, the vaccine hunter, Nelson Michael and Merlin Robb. And we had Ayavi, Reagan Institute with Galit Alter and her interesting tools to look at immunoresponses, the humoral immune responses in a different way, and more recently joined by the HVTN and the Gates Foundation. So next slide. Yes, we have an aspiration within Janssen to develop a prophylactic vaccine able to protect against multiple clades. So we want to develop ultimately a global vaccine. That's our target, that's what our aspiration. And in the early development phase and in preclinical, obviously, we tested different components in heterologous prime boost regimens. And a key component for us is the Ad26 vector, so that's alternative vectors compared to the Adeno5, and together with Adeno26, also POX vectors like the MVA, so the modified vaccine Ankara viral vectors, both delivering the mosaic inserts, which have been mentioned by Mitchell, and these inserts were designed to cover multiple clades and increase the breadth of immune responses induced by those vaccines delivering these type of antigens. And at least we have seen this and demonstrated this in in multiple preclinical studies, some of them published in scientific journals. And later on, also, we kept on adding components to improve the humoral immunity, and these components are trimeric envelope proteins, so-called GP140s, which we are now testing in the clinic as well. So next slide. Yes. So I did mention some of the proof of concept studies, and these were largely conducted in non-human primates, and with Dan Baruch and his team leading the preclinical studies. And the initial data were obtained using the vectors, so the viral vectors, not yet with the protein as a boost, and using heterologous prime boost regimens with Ad26 used as a prime and MVA vectors used as boost. We showed in two different NHP models protection against exposure, against acquisition either of the simian, so HIV virus, or the hybrid, so-called SHIF, simian HIV virus. So against challenges, repeated challenges with these different viruses, we demonstrated a high level of per-exposure risk reduction, above 80% in these studies. However, after the completion of the series of challenges, in this case 6 consecutive intrarectal challenges, almost all the animals did get infected. So already good results, encouraging results in terms of per-exposure risk reduction, but we kept on building on. And the next slide shows, summarizes the data published a couple of years ago now in Science, where indeed on top of the viral vectors, we included the GP140 proteins as a boost. And we did see first that the humoral responses were indeed increased in magnitude. We also saw that the functionality was improved in these animal studies, again using the two different models with SIV or SHIF. And in both cases, again, we reproduced a high level of per-exposure risk reduction, but this time in two separate studies. When we were boosting with GP140 animals previously primed with Ad26, either with the SIV inserts or the mosaic insert that we have now in the clinic, we saw that a significant amount of animals did not get infected. So we saw full protection after the series of 6 challenges, again, intrarectal challenges. So next slide captures the components that we decided to move into the next stage of development. So based on the encouraging results from these non-human primate studies, we decided to move into the GMP manufacturing, so manufacturing of clinical trial material, so material that would have a grade to enable us to enter into the first in-human studies. And while we were producing these materials and starting small first in-human studies with some of these components, we also decided to initiate a study in non-human primates mirroring exactly the regimens that we planned to test in a phase 1/2A study, and I will talk about this study later today, the study that we call our approach. So the study was designed to evaluate multiple heterologous prime boost regimens, where we always use the Ad26 component, delivering mosaic inserts as a prime at 0 and 3 months, or sometimes we refer to weeks, so 0-12 weeks. And then at 6 months and at 12 months, we come with differential boosting. So in some groups, we would boost with Adeno26 and at the same time the GP140, clade C, or the GP140 alone as a boost, or boosting with the MVA vector, so Adeno26 prime MVA vector boost, with or without, again, the GP140. So these are multiple regimens, and later on, when I will show you the approach study, we will really see all the different arms tested in humans. So if we go to the next slide and we see the outcome of the challenge study. So once again, we use the same shift challenge model used previously and published in the Science paper 2 years ago. And we were glad to see that multiple regimen. If you click to the next slide, we highlight those regimens with the highest per exposure risk reduction, with regimens that in this case all contained the GP140 boost, either alone or in combination with Adeno26 or in combination with the MVA in the boosting. Again, the prime is always Adeno26 at 0-3 months. And most interesting for us was the Adeno26 as a prime, followed month 6 and month 12 with a boost with Ad26 again, and the GP140 clade C also had again a significant, statistically significant full protection after the 6 challenges. So again, we confirmed previous data, and this time, importantly, we did use exactly the components that we were testing. We started to test in the human study. So we can go to the next slide now. To also very quickly summarize the findings in this study, where we confirmed that humoral responses and measured by ELISA, so IgG binding antibodies, as well as ELISpot2 envelope, did correlate to improved protection. And in addition, as previously demonstrated, we did see that functional antibodies like the antibody-dependent cellular phagocytosis were also found to have a good correlation with protection. So these are important findings, and I'm summarizing very quickly here since the correlate of protection analysis is almost still ongoing. So we went on also with other exploratory studies for this non-human primate study, so that we can also guide what we are going to look at in the clinical studies. So next slide. Here we start summarizing where do we stand, what did we do in humans. So when I mentioned we decided to do the non-human primate study mirroring the phase 2A study, at the same time while the monkeys were being immunized and while after immunization 6 months later we were challenged, we entered into first-in-human studies, and we decided to test some of these components as a standalone, since as you can see, we do have quite different components. So we did have the MVA mosaics into a small phase 1 study, first in human for MVA mosaic, but even for the mosaic inserts, and this was a study conducted at one single site in Boston at Brigham with Dr. Lindsay Baden as a principal investigator. And here we did see that the MVA mosaic vector were able to induce cross-clade ELISA responses, so were immunogenic and well tolerated in humans. We also had a small phase 1 study in US, in Florida this time, where we tested for the first time the GP140 clade C with or without aluminum phosphate, which is the adjuvant used in our clinic. And again, the clade C, when combined with alum was shown to induce antibody responses. Now, when we go to the next slide, here we finally have the phase 1/2a approach study, and this is indeed the first in-human evaluation of Ad26 mosaics, as well as all the heterologous regimens I mentioned before. This is a multi-country, multi-site study in US, Rwanda, Uganda, South Africa, and Thailand. We targeted 400 volunteers, and we have been the regulatory sponsor for all these studies so far. And we had many co-funders in this study, all listed here. And something to mention is that we decided to select a mountaineering theme for our phase 1/2a trials, and here you see what do we mean, what approach means in mountaineering terminology is the path route to the start of a technical climb. Although this is generally a walk or at most a scramble, it is occasionally as challenging as the climb itself. And we did feel we had some challenges. We were excited to do such a study, but it's not the simplest phase 2a study. So we go to the next slide, and you see a little bit what I mean. It's a study with 8 different groups. Group 8 with placebo. Each group we targeted to have 50 subjects, and we see group 1 with our lead candidate based on the preclinical data with Ad26 followed at week 24, so the month 6, and week 48 by Ad26 again as a boost in combination with the GP140, 250 micrograms. And then group 2 is pretty much the same regimen, however, testing a lower dose of protein, 50 micrograms. And then group 3 is a control group with Ad26 alone, no heterologous boosting here. And then group 4 and 5, very similar with this time the MVA mosaic as a boost, and again the different doses of GP140, 250 and 50 in group 5, while group 6 had only the heterologous vector as a boost, MVA mosaic alone. And finally, group 7 with the GP140 as a boost without any vector. So pretty much the same groups we had in the monkey study I've shown you, with the only difference that in humans we also had the opportunity to evaluate a lower dose of protein, the groups 2 and 5 particularly. So this study is still ongoing in terms of long-term follow-up. So we want to look at durability of immune responses as well. However, indeed, in all the groups, vaccination has been completed. So next slide. We went on while the study was enrolling and vaccinating within our discovery team, as well as the team of Denbaru. 2 additional components have been evaluated in preclinical, a fourth Ad26, the so-called Ad26 Mos2SM. So we added finally a second envelope, mosaic envelope, to our Ad26 product, and this in preclinical showed to increase the breadth of immune responses, particularly towards improving the clade C responses, while adding the mosaic GP140, which was developed by the lab of Denbaru, we saw again an improved breadth of immune responses with more clade B, so with clade B responses improved. So we thought that these findings were important for us, since we are aiming at a global vaccine. And these components are now being evaluated in 2 separate phase 1/2a studies, studying the same type of intervals. So again, the 26 drug product is given always as a prime, and in the boosting then together with the proteins. So next slide shows the first study, the so-called TRAVERSE study, Progress Forward by Moving Horizontally, where we are evaluating this new Ad26 MOS for HIV, so the 4-valent, and we are comparing it to the 3-valent Ad26, the one that has been used in approach. And after the priming with these drug products, we are always boosting in TRAVERSE with the clade C GP140. In this case, the study is running in US and Rwanda with 198 subjects, and we did start vaccinating in July 2006, last year, and I'm glad to inform you that enrollment has been completed smoothly, and vaccinations, boosting vaccinations are ongoing. The study is conducted largely in US at VTN sites and in Rwanda at a site that has been in the past also from IAVI. Next slide. And ASCENT HPX203 HVTN118 is the study where we are finally testing for the first time the full component. So the regimen which we hope to confirm is going to be the regimen for a global vaccine. So here we are testing Ad26 MOS4 as a prime, and the boosting is either with a vector again and the Mosaic GP140 with clade C GP140, so a bivalent GP140 booster, or with the clade C GP140 alone as a comparator. And the study started recently, so we have the first subjects vaccinated, and it's a study again multi-country in US, Kenya, and Rwanda with 150 subjects as a target. And next slide. A small ancillary trial ongoing, single site, this time at BIDMC, HPX102. We started last year, and in this trial we are investigating different vaccine schedules with the adeno trivalent and clade C GP140. It is co-funded by Regon, BIDMC, and Janssen. Again, with this study, we are not going to select a regimen for upcoming efficacy trial. However, we believe that evaluating different schedules, simpler or shorter vaccine schedule may be important for late-stage development. Next slide. So how do we move from a phase 2A to phase 2B and phase 3 licensure efficacy trials? We are testing multiple regimens. How do we select the regimen to then move into large-scale studies? So we have hypotheses. We hope and to have a vaccine that will induce balanced immune responses with both humoral and cellular immunity, and again, a global vaccine should provide broad coverage against multiple clades. And we have based many of our phase 2A studies on the findings in non-human primates. So besides the protection observed, we have been able to identify potential correlates of protection. So we give importance to those findings from the non-human primate studies. And obviously we also look at the immunocorrelates identified in the RV144 to correlate with improved protection against HIV infection. So next slide. We have regimen selection, but most importantly, we have go-no-go criteria to move into these large-scale trials. And obviously, we will look at vaccine take. We are looking at the percentage of responders with multiple assays, looking at the humoral and cellular compartments, and we look at functionality of the antibodies because, again, in our preclinical studies, functional antibodies, non-neutralizing but functional, have been demonstrated to correlate with improved protection against acquisition. We have multiple decision points based on data coming from the approach study after the third and after the fourth vaccination, as well as after Traverse interim data. So altogether, we have agreed with our partners on go-no-go criteria, on a package of data that we hope will confirm we are on the right track and to start a proof of concept study later this year or early 2018. We aim at later this year if possible. If all the data will confirm we have the right vaccine in our hands to start a proof of concept study. And next slide. This, I think, is almost my last slide before the acknowledgement, and this is what we are planning with our collaborators at HVTN. So we have a study HPX208, HVTN 705, which is also posted in clinicaltrials.gov with more details. So the idea and the goal is to conduct a study, placebo-controlled, double-blind, in Southern Africa, South Africa, Zambia, Zimbabwe, Malawi, and Mozambique, targeting 2,600 volunteers, and these are sexually active, HIV-negative women aged 18-35 years old. So again, as I mentioned, it's a one-to-one randomization, so 1,300 women will receive the vaccine and the other 1,300 will receive a placebo, with the intervals as mentioned before, so our classical vaccine regimen with vaccination at 0, 3, 6, and 12 months. And we will look at also durability if we pass the first stage. So after 2 years is the so-called stage 1 ending. It's the primary analysis. However, if we are not stopping for futility, which we all hope is not going to happen, then we will proceed to stage 2. So that we will improve our knowledge on durability of efficacy. So next slide. Oh, this is already the acknowledgement. So obviously I mentioned before the institution with whom we are collaborating, and here we have some of the key collaborators, but many, many more. All of them have enormous teams working and collaborating with us. So, of course, I mentioned Dan Baruch. We have the Gates Foundation, and importantly, Mike Pensiero, Mary from DAIDS, and from the HVTN, the team of Larry and Glenda and Julie, and we have Ayavi, Betty Korber, who designed the mosaics, and the MHRP with Nelson and Merlin and Julie, and finally Galit and Bruce Walker supporting from the Reagan Institute. And next slide shows some of the names from our team. We have here the compound development team leader. Frank Tomaka is our clinical leader, and Hanneke, Melanie, Johan, and Posthoff as our senior leadership who have been supporting us for this program. And many, many thanks to all the volunteers for their participation in our clinical studies. And this was the last slide.
I am so sorry, Maria. I was saying how great you were, and I was talking to myself on mute. I apologize. That was a terrific presentation, as I was saying to myself, with a lot of information and just a really exciting program that you've described. And a lot of questions have come in that I have been collecting here, and people can continue as you see on the screen. You can put them on the Ready Talk feature, and I will put them in the queue. People can still keep sending to avac@avac.org. And in a moment or two, after a few questions, people can unmute their line by hitting *7. I would ask you to introduce yourself and ask your question briefly, and then *6 again to keep the line nice and clear for the recording. But again, Maria, thank you so very much. And a lot of questions spanning a number of different issues, but one that came in that I think is really interesting, and you talked about a number of times the importance of finding a truly global vaccine, one that would work in different places. And going back many, many years, there have been conversations about the various clades and subtypes and will they matter. And a question came in from a great advocate saying, why hasn't the multi-clade approach been used from the beginning, since we always knew there were these different clades? And what's different and what prompted this program to really focus on that multi-clade approach, and why is it not the standard?
That's a very, very good question, and that I can think about many things, but one, it can, we don't know, how potent the mosaic will be, right? Some people have been concerned about, inducing, about dispersing, right, the immune response, and maybe by having so-called, let's call regional vaccine, then one could think, we focus on that, we will have the most potent responses, versus, asking too much from a component. So again, we don't know who's right, but we believe that you know very well how many years you have seen, for how many years we have been working on developing a vaccine with our collaborators, and we believe that if we are able to reach this goal with the mosaic components and have finally a vaccine that will fit all, then we are there, right? And we don't need to repeat this for many years to come. So again, we don't know yet, because we do not have efficacy in humans, but that's the aspiration that we have.
Great. No, incredibly helpful. And a related question in terms of the geography of the trial. And you described, and one of the big messages I think we all have really embraced is the need for these global partnerships, both in terms of the various organizations you mentioned and the geographies. And so someone asked a question related that there were trials done in many different parts of Africa with different subtypes, and there were studies also done in the United States. But a question, as you presented at least to the design for the potential HPX 2008 study, the efficacy trial, that it's just East and Southern Africa, and someone asked a question, what about the U.S.? Would it not still be a relevant place for efficacy?
Absolutely, absolutely, and that's what we plan to have in the near future. However, what we decided to do for US is to wait until we have the data from ASCENT. So if you recall, the ASCENT trial is the one where we are boosting not only with the clade C, but also with the mosaic GP140. So the idea is that as soon as we have, the safety data as well as the immunogenicity data from ASCENT, then we will decide, hopefully, we will have a go to also explore the efficacy of this combination in US. Because what I mentioned, and again, I realize that there is a lot of information and it's complex, but I mentioned that by adding the mosaic GP140, at least in the preclinical models, we did see an increase in the clade D responses, which are relevant, right, for a study in US. Again, already without such a GP140, Mosaic GP140, we do induce. We see in the preclinical, and we saw it also in our first-in-human studies, we induce responses against multiple clade because the mosaics are priming, nicely priming. But we saw even an increase in magnitude when adding. So yes, US, it's definitely in our plans, no doubt about it, for efficacy.
Great, and I should have said Europe too, especially given that you are a European and based there. So, but clearly a desire for global. I think one thing, and it was actually more a comment that had come in. I think one of the things that your presentation really represented is that the historic view of vaccine development, phase 1, phase 2, phase 3. Is never as clear and linear as people might hope and want. And I think what you're describing is the multiple phase 1 and 2As, and it's not just a single pathway, and you have lots of different studies, each informing the other. And I think that that's a really important point, that it is more complex than we might have imagined in the early days of AIDS vaccine advocacy. So super helpful. Speaking of, another question to come in, speaking of early days and history, many people hear, and someone asked a question about adenovirus vectors. And so many of the advocates on the line have long memories and the experience with two different studies of the two different adeno5 vector-based vaccines, the Step and Pambili study with the Merck Ad5, and then the HVTN 505 with the Vaccine Research Center's Ad5. So what's the difference, as someone asked here, between Ad26 and Ad5, and is there any reason to be concerned given past experiences with Ad5?
Yeah, fair question, absolutely. So what we have been doing from the very beginning with within our collaboration with Dan Baruch and under IPCAVT grants was really to look for alternative serotypes. And this search started because Ad5 had a very high seroprevalence, not only seroprevalence, but high titers against the vector. And we did see that the vectors, these serotypes are different. Yeah, they are different serotypes indeed. They are very different. They have different biology. They have different receptors. And I believe Dan wrote an interesting review describing also how different is the immediate response to these two different vectors, 5 and 26, the different inflammation profile, and so on and so forth. And when tested in preclinical, so not only in terms of biology, but when tested as vaccine vector for HIV in preclinical models, Ad26, when Ad26 was used, we have been able to achieve significant per-exposure risk reduction and in some cases also complete protection in a large amount of animals. This was not the case in the same models when adeno5 was used. So also in terms of efficacy in preclinical, obviously we do not have efficacy data yet with Ad26. Ad26 is shown to be a superior vector. So we believe there are enough differences to move ahead, and enough differences and enough promise from our preclinical studies to test this combination with the vector and the protein. I mean, we know that Adeno26 alone, without any protein, at least in the animals, does not show any real efficacy. So it is the heterologous prime boost regimen that makes this efficacious, at least in preclinical, and we hope to confirm it in humans.
No, that's great, and a really important point from the scientific side of the difference between the adenos. An additional question came in that relates to that, and that is how we, and I think the we being not just Janssen and its partners in the program, but all of us working in the HIV vaccine community, how we should be preparing trial communities for this next efficacy trial. And there are two parts to that question that someone asked brilliantly. One, how are we preparing communities for this trial, particularly in places where other efficacy, both vaccine efficacy and non-vaccine efficacy trials are happening? And the second part was particularly in South Africa, which of course is hosting many of these trials, including the HVTN 702 trial following on the Thai trial, but also which obviously hosted the Pambili study with the Merck Ad5. And so a question of how do we prepare communities to understand what's coming next, how it fits in with the current studies, and how it relates or doesn't, as you just described, to the past ADNO5 trials, and wondering how Janssen is planning to work or prepare trial communities and engage.
Now that's a great question, and again, the trial, we hope to start the trial, and we do hope to start this trial this year, pending confirmation of all the data that I mentioned before. So assuming that we will successfully have a go, there are initiatives that are being kicked off very soon. And again, Janssen is not doing this alone. So we have our collaborators with the HVTN. So there will be already in the coming weeks and months exactly initiative and workshops organized in the countries, especially on communication, engagement, and stakeholders. And some of you, right, will also be involved in communication and alignment. So we and within Janssen, Michelle, we also have pretty much carry on workshops on GPP, and it's a very important topic for us within the company. So again, will be a joint effort with our collaborators, but it's going to start assuming that we will have a go very, very soon.
Great. No, very helpful, and I will say it is incredibly exciting and heartening when you describe the company's commitment and engagement with GPP, and I hope everyone on the line knows about good participatory practices, and really exciting the way the company has embraced it and really tried to operationalize it. Thank you and your colleagues for that. You mentioned something in that answer, which was terrific, about you said something, assuming all of the trials lead to the go decision. And there was a question from actually an advocate in Europe asking about, and I think a couple of people commented on how they loved the corner slides where you have the ascent and the traverse, and clearly you are all leading a climb up a very large mountain here. But a question about the way in which, what is kind of triggering and leading to what. So a question about the difference between approach and ascent, and what's kind of determining the large-scale study. So if you could just help, and I can maybe go back to a slide that might help us understand where ascent fits in, because on the slide that I've just put up, you show very nicely approach to traverse. So I wonder if you could fill in the blanks there. Yeah. So indeed, very good point, because the arrows here below are really showing, the blocks of data needed for us and for our partners to agree, yes, we have the right regimen for the proof of concept. So not so much for the large phase 3 global study. And I will explain that. So again, let's go first to the POC. For the POC, as I explained, we are going to test Ad26, the 4-valent, so assuming the 4-valent, which is tested in Traverse, we are testing Ad26 as a prime, at a 0-3, and as a boost also 6 and 12 months in combination with a clade C. That is for the proof of concept. The regimen, so the intervals that we are going to test in the POC are the same intervals that we are testing in approach, right? So an approach week 28, what does that mean?
the week 28 is after the first protein boost. And based on all our preclinical studies, we see that already one boost of protein brings very high immune responses, both humoral but also cellular immune responses. Those responses that in the monkeys correlated with increased protection. Then we have approach week 22, and this means we are looking at the second boost with the protein and see whether we are at least maintaining or increasing further the immune responses. This last immunization is planned to, with the aim of increasing durability of immune responses. And then the TRAVERSE is the study that shows that is testing for the first time the 4-valent Ad26. So this study approach and TRAVERSE are testing the components that we are planning to have in the proof of concept. Then for phase 3, for an efficacy study that eventually will run in the Western countries, in US and European countries, then we need the other study. The study that is not here, but I explained earlier, is the so-called ASCENT, because there is where finally we are also including a second protein in the boost. And that's the study that we are going to use to finally confirm and define the regimen for the global efficacy studies, not proof of concept, but eventually phase 3/licensure studies. I hope this explained a little bit or clarified.
No, it does very much, and I think putting those pieces together, and I think it goes back to this issue that as much as we all try to boil things down to the simple, phase 1, phase 2, phase 3, that a program like this to get to licensure, and I think we saw that with the ALVAC protein boost with Thailand now into South Africa, that none of this is a simple, clear, linear path, and there's lots of components happening simultaneously, so incredibly helpful. There are some other questions that have come in, but before I realize we're getting close to the top of the hour, before if we have time at all for those last couple of questions, let me just pause for a moment and see if anybody has a question on the line. Again, you can hit *7 to unmute your line and just introduce yourself, and then *6 to remute. Any questions on the line? Okay, because people were very dutifully sending questions as we went. Incredible. One question that came in that I think important to ask, you had mentioned that you'd be looking for functional antibody responses in particularly the next trial. And someone asked a question, can you provide a really brief overview of what kind of responses you're looking for?
Yes. So, based on the non-human primate studies, we did see that some of these functional activities, as particularly the antibody, so the ADCP, antibody-dependent cellular phagocytosis, did have a positive correlation with increased protection against acquisition. So we have not, we know that with our regimens we are not going to induce the holy grail, so-called broadly neutralizing antibodies. But there are other functions that antibodies have and appear to also, have a good effect, a good protective efficacy in at least in the non-human primates. We will also look at other functions like ADCC. We will look at, whether we like for RV144, we do have IgG3 responses, but particularly based on our non-human primate studies, we would like to explore these other functions that we have been looking at together with the labs of Galit Galit Halter at MIT.
Great. Maria, thank you. That was a terrific answer and incredibly helpful. And as you know, and as we talked about before the call, we have a mixture on this line of people who work in vaccine science and in HIV science generally, and many more advocates, and your ability to convey what are very complex issues so clearly is just terrific. And I was, you mentioned just at the end there about the different antibody approaches. I just, on the slide, if people are still online, you'll see the lineup for the next 4 in the series that Maria has kicked off today that will actually begin to fill in some of the other pieces of this incredibly diverse and exciting scientific pipeline of activity right now. And it will include conversations later in May about the HVTN 702, the actual efficacy trial that is already underway. And then you'll see on either side of that conversations with the Vaccine Research Center, as well as Lynn Morris from South Africa, talking about neutralizing antibodies that Maria just mentioned. And then finally with Nelson at the end looking at vaccines broadly. And I just want to say that the whole series of calls, but particularly Maria, your talk itself really highlighted a few things that we really believe at AVAC guide us right now, 20 years since President Clinton described the importance of an AIDS vaccine and prioritized that. And in fact, in that speech in 1997, it called out the development of the Vaccine Research Center. So this whole series of calls in your talk really describe both the essential need for an HIV vaccine still, and excitingly you talked about that it's possible, and the evidence through certainly the non-human primates and early clinical development that shows that this is possible. You showed us, I think, very clearly the progress within your own program and how it fits into that broader pipeline. And finally, I think just want to thank you because as you said many times throughout the presentation, you named a whole number of partners, a veritable alphabet soup of acronyms of organizations working with you, coming from the nonprofit world, coming from the U.S. government, obviously the industry role that Janssen is leading, and the philanthropic support as well. And one of the key messages certainly that is quite different today in 2017 than it was in 1997 was this global network of partners collaborating, and not just on the scientific side, but as you so nicely pointed out, the importance of community engagement and communications and stakeholder involvement, which we all know is fundamental to the scientific pursuit of an HIV vaccine. So it was, I'm so pleased that we were able to kick off this whole series with you because you really highlighted not just the Janssen program and the Ad26 program, but what I think it is safe to say are the key messages that not only are important in April and May 2017, but really guide our collective work. And just really want to thank you again for spending time with all of us, and looking forward to many more opportunities as the program moves forward.
Thank you, absolutely. Thank you all.
And thank you all for joining again. We will have our next call on May 4th, which is next Thursday. And it's actually very exciting because, as I said in the speech that Bill Clinton gave on May 18, 1997, he specifically said, we had to do more and better and faster, and to do that, the need to create a vaccine research center at the National Institutes of Health was announced, and so we'll have Barney Graham from the VRC describe both the history, but perhaps more importantly, the future as well, and then the other calls to come after that. Thank you all again, and please do keep those questions coming in for Maria and for others on the webinars to come, and look forward to working with you all going forward.
Thank you. Thank you again.
Cheers. Have a great evening, Maria. Thank you again so very much.
Bye-bye.